Today we will see the another unit, that is the unit number 3, chromosome structures and variations. Under chromosome structures and variations, we will see the multiple allelism, introduction, definition and self-incompatibility in plants. First of all, we will see the multiple allelism. A gene has two alternative forms called as alleles. Usually, one of two alleles of gene is dominant over the other, which is a recessive. The two alleles of genes determine the two contrasting forms of single character. For example, round and wrinkled seed shape in a pea, but in many cases, several alleles of single genes are known, each governing a distinct form of concern trait. Such a situation is known as multiple allelism, and many alleles of single gene are called as multiple alleles. First, we see the self-incompatibility. In many species, the active and functional pollen grains fails to effect the fertilization on self-pollination. As a result, there is no seed set following the self-pollination, but the same flowers produces seeds on a cross-pollination. This phenomena is called as self-incompatibility. It was first described by Coelho. In a self-incompatibility, the pollen grains fails to germinate or enter the stigma. The pollen tube may grow too slowly in the style to effect the fertilization before the flower drop or the embryos degenerate. The genetic control of cell compatibility is a complex. Lewis in 1954 has suggested a simple classification of self-incompatibility. That self-incompatibility is divided into a gametophytic system and sporophytic system. First, we see the gametophytic system. In this system, the self-incompatible reactions of pollen grain is determined by their own genotype. This is because the substance involved in a self-incompatible reaction of pollen is produced after meiosis. In a style, the two S allele shows codominance. In many species like Nicotiana, Lycopersicum, Solanum, Petunia, the self-incompatibility is specified by a single gene, that is the S, with multiple alleles like S1, S2 and S3. In such cases, the polyploidy may be a eliminate self-incompatibility. In some species like Saleles, two loci, that is the Z and S, controls the self-incompatibility, while in some others like sugar beet, papaver, three loci are involved. In these cases, polyploidy has no effect on self-incompatibility. The mutant alleles, that is the SF of S. locus, allows the pollen grain carrying it to effect the fertilization of flowers with any S genotype. The pollen Si reaction due to its own genotype at S locus. Generally, one gene with multiple alleles, that is the two or three genes also known. The S allele shows codominance in a style stigma surface weight. The pollen tube growth retarded in a style. The self-incompatibility is suppressed by polyploidy or X-ray or irradiations. Or you observe the picture. In a picture, there is a genotype of plant, that is the sporophytes, S1, S2 and S3, S4. The genotype of pollen, that is the S1, S2, S3 and S4. The incompatibility reaction of pollen grains, that is the S1, S2, S3 and S4. The incompatibility reaction of style is S1, S2, S3, S4. While you observe the first picture, there is a S1 and S2 pollens, and in a stylar region there is also presence of S1 and S2 alleles. In such cases, the fertilization is not takes place, or there is a retardation of pollens. It means it shows a fully incompatible. While you observe the middle picture, in middle picture, the pollen shows a S1 and S3 alleles, while in a stylar region, there is a presence of S1 and S2. That means that S1 and S1, which is a present in a stylar region, it shows codominance. The another S2, which is a present, and that S2, which is not uh, compatible with that S3, that means it is incompatible, and that's why the S3 in which the germination takes place, that means it shows a partial incompatibility. While you observe the third picture, in a pollen grains there is a presence of S3 and S4 alleles, but while in a stylar region there is a presence of S1 and S2 alleles. It shows a fully compatibility. That means the germination of both pollen grains shows. If pollen grains lands on a stigma which has a S same S allele as the pollen, for example S1 pollen on S1 S2, S1 S3 and S1 S4, etc., that is the stigma, it germinates normally, but the pollen tube growth is so slow that fertilization does not take place. However, if the stigma does not grows normally and fertilization takes place, therefore, depending upon the S locus genotype, the mating between the different plants are of three types, that is the fully compatible, partially incompatible, and fully incompatible. This self-incompatibility first discovered East and Mangels in 1925 in a Nicotiana Sanderi. They finally concluded that the pollen grains do not germinate on the stigma of same type of the plant was due to antigen-antibody relationship. The tissue of stigma and style produces the antibodies to counteract with the antigens of pollen grains of same genetic makeup. Thus, the reaction antigen antibody stop the germination of pollen grains and growth of pollen tube. The second type, that is the sporophytic system. This system was first reported by Hughes and Babcock in 1950 in a Crepis foetida and by Gerstel in a Parthenium argentatum. In this system, the SI reaction of pollen grain is determined by the genotype and S locus of the plant which produce it. The self-incompatible reaction is controlled by the single locus S with multiple alleles. The S allele shows a dominance in the following manner. That is, the S1 dominant over the S2 is dominant over the S3 and S4 and S5, so on. Therefore, the plant with genotype S1 S2 will produce the pollen grains of single self-incompatible reaction, that is the S1. The stigma of its flower will also have the self-incompatible reaction S1. On self-pollination, the pollen of S1, that Si reactions will interact with the stigma cell of the same S1 Si reaction. These interactions generally prevents the pollen germination or entry or the growth of pollen tubes in the stigma. But when S1 S2 plants, these are the female, is crossed with S2 S3 plant, which is the male, the pollen grains with S2 Si reaction is placed on the stigma of different S1 Si reaction, pollen germination, and pollen tube growth. As a result, the normal fertilization takes place. The S allele shows codominance or competition. The stigma surface, which is dry. Usually, the pollination occurs when the pollen grains are trinucleate. Incompatible reactions of pollen grains appears due to some compounds of tapetum being deposited on the egg site. The pollen Si reactions due to S locus genotype of plant which produce it. A single gene with multiple alleles. The S allele shows dominance, codominance or competition. The stigma surface is dry, covered with a pellicle. The incompatible pollen induce callose deposition in stigma cells. The pollen germination tube entry in a stigma is inhibited. While you observe the cross, that is the genotype of plants S1, S2, S1, S3, S1, S4, S2, S3, S2, S4, S3, S4, the genotype of pollens like S1, S2, S1, S3, S1, S4, S2, S3, S2, S4, and S3, S4. The incompatibility of the reactions of the pollen grains all S1, all S1, all S1, all S2, all S2, all S3. The incompatibility reaction of style S1, 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 S2, S2, and S3. Pollen genotype S1, S2 while interacts with the stigma of S1, S2, the completely incompatibility, that means the S1 and S2, which is the self one. I'll observe the middle picture. The pollen genotypes, these are the S1 and S4. While in a stigmatic region, there is also presence of S1 and S2, completely incompatibility. While you observe the third picture, the pollen genotypes, that is the S2, S3, and interacts with that S1, S2, there is a germination occurs. That means the completely compatibility is seen. Thank you.